Hello, chat. Welcome on in, everyone. How are y'all doing today? Look at that perfect timing. Just as the prediction ended is when we came on over here. How is everyone doing today? Yes, Music Girl, ask your question. You shall receive a potential answer. Chat, let me welcome everyone in. I have totally nerfed and missing the very top of the chat. I apologize for that, but I did see Smikes was first. Smikes was first. Yes, please do, Music Girl. Just don't stab me with it. It looks like a stabby stab. Guys, Music Girl's in his house. Smikes was first. Lady Lara Croft. Detector. Lady Lara Croft coming on in with the 130,000 channel points to receive the shiny Lady Lara Croft. That shiny looks very, very good on you, Lady Lara Croft. Hi, NMD96 Milas. How are you doing, Milas? Welcome on in. Debs. Hello, Debs. How you doing, Debs? Andrew, say. Hello, Andrew. Supernatural Writer. Hello, Supernatural. Cliff, hit it! Hello, Grimly. Hello, High Tech Redneck Dev. High Tech, how you doing tonight? High Tech, it's great to see you. High Tech, guys, go, lovely, amazing. High Tech. Supernatural, how you doing? Music Girl, yes, you can bring your stabby. You can bring your stabby. Music Girl. Nerdy Knots, hello, Nerdy Knots. How you doing, Nerdy Knots? It's great to see you. I know you don't like the angry ants, but it's gonna be okay, Nerdy Knots. That was requested by, very much so, Gwendolyn P. The lovely Gwendolyn P. Debs, did you have the chocolate? Did you have the chocolate, Debs? What kind of chocolate did you end up having? Please just don't stab me with it, Music Girl. That will do. Don't stab me. Don't stab. Goes up by 5K, Gwendolyn P.
It's alive! I've always wanted to say that.
Look at that. New animation right there. Let me see. Let me catch up on chats. Let me just... Travel the World. Hello, Travel the World. Welcome on in. Let me see. We also have DMX. Hi, DMX. We also got Epico Sam. Epico Sam, how you doing, Epico Sam? Welcome on in, Epico Sam. Right, there we go. Debs. Becca, hi, Becca. How you doing, Becca? Cliff, the lovely Cliff, and the lovely High Tech, and the amazing Grimly. Look at those amazing people. Detector, how you doing today, Detector? Let's see. Food and drink status, indeed. Hi, Odin. Odin, I am a real scientist. My PhD is in molecular and systems biology, and I am a research scientist indeed. Welcome on in. Yes, Lady Lara Croft, exclamation point Amber for the new, Odin, thank you for the follow. Welcome to the colony. Exclamation point Amber, Lady Lara Croft for the Amber animation, and exclamation point Rage.
Wanted to say that.
And then the rage for Gwendolyn P. The rage for Gwendolyn P. Hi, Chris TS.
Gucci and the Hoochie Mama.
And we do have a new Gucci tonight. We do have a new Gucci Travel the World. Lady Lara Croft submitted her Gucci, and that Gucci is in fact live tonight, Travel the World. So now we're up to, I think, 42 Gucci's. It looks just like Gwendolyn P. Gucci. Thank you, Smikes. I was hoping, Smikes. Actually, Smikes, listen. Smikes, what I think I'm going to do actually is go back and animate a little bit more on it. I want to tweak a little more and randomize there being one rage where the fire is obscenely large. Where she creates such giant purple flames. It'll maybe be like, 10% chance of likelihood. But then I just want it to be just like epic, powerful flames. Like, I think the muted one is better for like the standard animation, right? And the standard emo, but just like something obscenely way too much fire where it's not even, doesn't even make sense anymore. Like, exactly, like just like a phoenix, Mike. Just like a phoenix. Becca, I'm doing alright today. Becca, I'm going to the post tomorrow. I know I have something at the post waiting from the lovely Becca. That is also live on the Discord now. So if anyone needs to show off a rage If anyone needs to show off a rage at the Discord, you can now do so. Thank you, Debs. Thank you, Debs. I needed a Debs. It's a little bit. There's a lot of bags coming, Debs. There's a lot of bag coming, Debs. There's a lot of bag. I'm glad the stickers arrived at Epico Sam. That's right. Epico Sam got one of the Vitruvian Ant stickers because Epico Sam did an art piece for the Antonia in Your Style art challenge. And Epico Sam, it's always lovely. Risto! Hi, Risto. How you doing, Risto? Welcome on in, Risto Rock! Risto Rock, indeed. How you doing, Risto? Where do we go find royalty-free sounds? Lady Lara Croft. So, chat, Lady Lara Croft got her VIP badge tonight. Lady Lara Croft, you actually, by the way, ma'am, you get, so listen, Lady Lara Croft, you get one for your VIP badge. You get a second one because you have tier 2 sub, and you actually get a video command because you're tier 3. So Lady Lara Croft, you actually owe us 3 different sounds that you would like. 2 sounds and 1 video. 10 seconds or less. Lady Lara Croft, if you wanted something like a particular sound from a song, that definitely has to be 10 seconds or less. A video clip, it could be from, you know, a very short clip from a movie. There's no pressure. Lady Lara Croft, you don't have to decide now. But I'm just letting you know that you have that many choices. You have, you know, VIP, tier 2, and then the tier 3 video. You got all the things. Currently studying mathematics as Odin. I would like to know how mathematician can contribute to research medicine such as treatment for cancer, diseases in general, or is it more of a statistical side? Calculation of the molecules, how it works, stands for your content. Thank you for the lurk, Sputnik. So, Odin, there's actually a lot of different ways that mathematics is very heavily involved. One is, as you said, the statistical side, and stats are extremely important. That's the way we interpret all of the data that does come in. And if you think we look at it, Odin, though, I will say there is an entire field now emerging of biological engineering. And what I mean by that is it's a series of engineering. It's breaking down the processes within a cell in a disease state in terms of an engineering language and circuitry. Meaning that, you know, if this circuit is on, then this other circuit will continue to be on. But if this circuit interferes with that, then the whole circuit gets turned off. So think of it like almost an interactive map of like electrodes connecting to each other, and if there's a lacking of signal between 2 electrical nodes, that messes up the signal between the 2 wires. And so there's a whole department at Dartmouth now, for example, that's evolving based on this front. It's again a mix of electrical engineering and biology, and there's a lot of mathematical modeling of that. When that course was being offered at Dartmouth, that was like a calculus based course, but it was primarily in biology. And again, these kinds of mathematical models are huge. Thank you for the lyric, Crystal! On top of that, Odin, there's a whole field of bioinformatics. Bioinformatics is when you're analyzing raw data, huge data sets. For example, RNA sequencing data, genetic sequencing data. You're analyzing to find differences and similarities between data sets, how much a gene is turned on or off in a normal cell versus a disease state cell, and all the combination in between. So there's a lot of overlap between mathematics, like just a pure mathematician, statistician, and a pure like biologist working with an animal, let's say. There's a huge level of overlap. Becca says, my mom didn't know I sent you the painting at all. I told her I was going to Charles, you were being... Oh, to the grocery store. I gotcha, Becca. Very cool, Becca. Did you show her a picture, Becca, though? Did you show her a picture? Hi, Bentley. How you doing, Bentley? Welcome on in, Bentley. It's great to see you, Bentley. Guys, a couple more shout-outs. We do have the lovely and amazing, breathtaking Bentley in the house. Guys, go check out the amazing Bentley. Hilarious content. The lovely just chatting and occasional gaming as well. Very spooky. Very, very spooky game sometimes, but, you know. But, yeah. A mathematician in the house. So, yes, it seems like Gwen. Lita is also very big on the math. So Odin, my wife and I run the channel. She is very big into. And you really look over Sam into the mathematical modeling side of science. So a lot of her work, in addition to being like working with the animals, is bioinformatics. So let's draw out a model. Why don't we just draw out a model? Because that makes it easier for me to explain these things. All right? Right, let's draw out the model. Models are always easier, chat. They're always easier. I know Detector. Detector loves math. Detector loves math. So, Odin. Very, very briefly, in a super non-in-depth kind of way, what you can end up doing, right, is there could be an interactome. So you can have cells, and we're actually going to be talking about this, 2 kind of cell data sets. One that are wild type cells or normal uninfected cells, and one that have cancer of some kind. And one thing that you'll find in cancerous versus normal cells is that there are multiple nuclei in cancer cells versus not in normal wild type cells, which means there's already going to be differences in gene expression. Right, so gene expression means how much of a certain gene is turned on or off. Is there a lot of RNA of that particular gene and a lot of protein being made? Or is there very, very little? And presumably there's going to be differences in between these cases. So you can take a look and see, maybe we can see, all right, this normal cell has gene, actually let's just go for beta-catenin. Beta-catenin is an important regulator in cancer cells, in normal cells, preventing cancer. It prevents overgrowth of cells. So presumably you'd have high levels of beta-catenin in the wild type cells, and in the cancer cells, you'd have low levels of beta-catenin. And then you could figure out what is the map in between that. So Odin, if you were wondering what is the interactome, so you could see, okay, you have beta-catenin, and that affects downstream growth factors inside the nucleus. This is then turned on by a molecule called Wnt. This is then inhibited by a molecule called APC, and so on and so forth. So you can build out these maps, and with these maps, you can figure out the level of signaling of each of these genes between your 2 data sets, between the wild type normal cells and then the cancerous cells, and have those comparisons in between. And then you can build these kind of circuit maps as well. Does that make sense, Odin? Does that make sense? Please to tell me. Gwendolyn P makes ice cream with her pectorals. I love it. I knew that making ice cream with your pectorals is nothing insane. I knew that this is 100% a all-around, like, this is always happening kind of thing. It makes sense? Odin, yeah, feel free to ask any more questions you have. Again, the mathematical modeling I did. Dabs makes ice cream with... I love it. I absolutely love it. So Odin, one thing that I did, this was the mathematical modeling that I did, and maybe this is more Odin, you're up your alley. So let's say you want to discover a new gene that's involved in learning and memory. I'm using that example because that's what I wanted to do, but essentially you can do any process that you're interested in. Hello, Suitable Substrate. Welcome on in, Suitable Substrate. So Odin, let's say you want to do a new learning and memory gene. So what you do is you go to your data set. You go to your data set, and this graph is going to be telling us gene expression. So this is that's what we're measuring. And for learning and memory, and you look in the data and you find you 6 genes that are involved in learning and memory. Okay, these 6 in red that we know. So we'll even write it down. Red, known genes. Okay? And what you can actually do is you can build a mathematical model to look at the relationship between these genes and find other genes that behave in a similar way. So you want to find genes that are also upregulated or turned on, similar to this group of red genes instead of this one. So here in our six genes, we have 5 that behave very, very similarly and 1 that's very different. And you maybe want to find other genes. Let's mark them in blue, or we'll call them candidate genes. You want to find some that fit here and not so much here, right? And those upper blue ones might be genes that have similar behavior to what you're interested in learning. So how do you actually filter something like this? So you have to run what is called an SVM model. An SVM model, I forget what it stands for because acronyms run me up the wall and sometimes it just doesn't hit. I will look it up what it means. However, the principle is pretty straightforward. Essentially, the algorithm draws a line in the center of this graph. And the axes are gene expression, so this is time in a cell, is the X, and then gene expression is Y. So low amounts of time within a particular cell, and certain genes, so this is multi-dimensional map, so it's like this gene is on for a low amount of time within this cell versus these genes are on high in this cell type for a long time. So it's looking at a couple of different factors. But you run this line through the center, and then you analyze distances of these genes to the red line. And these distances inform the model of what it's looking for. So what you'll find is these distances... Love you, Smikes. These distances here will average a certain length. And then what the mathematical model does is it says, okay, I am going to find other genes on this side of the red line that are roughly the same distance from this red line. So we might find a couple of new that are also at a similar distance. It doesn't tell you that it's necessarily a learning and memory gene. It's just saying these genes are behaving similar to the ones that you told me you were interested in. Champ, that brain of yours. I don't. Oh, Smikes, you're working on the algorithm. Thank you, Smikes. You're breathtaking, sir. You're breathtaking. Thank you for calling me Champ. Makes me feel special. Makes me feel lovely. So Odin, chat, y'all feel free to ask me any questions. I know this is kind of a deep topic for just kicking off the stream. Does that make sense, Odin? So what you do is you end up getting a list of genes that behave similarly to your known genes. So your list might be, a red gene, a red gene, a red gene, and maybe in between those gene levels we got a purple gene, and then we got a blue gene in there as well. And so then your red results. You know that these results, yes, those are what we want it to be. Those are learning and memory genes. But then you have to test. We got to test our blue gene, and we got to test our two purples. And that's when you take it back to the lab. So then you test out and see, do these new genes behave the same way as your old ones? And that's how you can find new genes that you otherwise may never have found. It's very... So Odin, I did this with fruit fly work. There are a lot of genes in the genome in the fruit fly that are unknown. They're just labeled with their CG identifier. So all the genes have either a gene name if you have identified what it does, or it's just, we know this is a gene, we don't know what it does, so we call it a CG gene. It's just a numeric combination that's given to it that allows it to identify, be identifiable on a database. And so we tested a couple of these CG genes that look like they behaved in a similar way to our known learning and memory genes, and sure enough, not all of them were learning and memory involved, but some of them were. It gets a little hairy, Odin, because the way you test it is you mutate the gene. And if you mutate the gene in the adult animal, you can have a couple of things different happen. One is you can have a developmental defect. And if we're looking at brain activity in these animals and there's a developmental defect, you can't say that it's really a learning and memory defect, right? You'd be saying, no, it was a developmental change. And so you have to analyze... you can control the genetics temporally So we're just adding more and more to it. Hi, Crystal. Welcome in, Crystal. So instead, if you have your adult animal, your adult fruit fly, and you mutate your adult fruit fly, so we do minus. That's the gene. Hi, DMX. Welcome on in, DMX. If you have that minus, that means it's a full mutant. And that means something could physically be wrong with the animal in its brain. Like, maybe a structure didn't form correctly.
Gucci
indeed. And therefore, if you tested it in your learning and memory assay, maybe it doesn't remember, but is that because its brain isn't normal? The other thing you can do is you can let the animal develop normally. So it's plus over plus as an adult. And then what you can do, you can actually go into its brain and turn off the gene in either all neurons or a very small subset of neurons. It is bananas. In fact, it's fruit fly banana. So that's the brain. Well, we can draw a better brain than that. So there's the brain, there's the optic lobes, there's the learning and memory center. Boop, we got some structure in there. You can turn off that gene in a particular series of neurons, and then you can ask if the actual effect that you observed is because of developmental mutation or because of actually neurological process. And that's how it's all due to this original list of genes that you've identified. And actually, let me grab the name of that model. Hold on, we can actually run the analysis if you would like. Unless it could have been shut. Oh no, maybe it's not shut down. Does the server still exist? Yep. The server no longer exists. However, I can show you all the original one. So here's the original server. This was the database that we built, and it's called Pilgrim. It's an interactive data-driven discovery platform for experiment scientists. It essentially does everything that we were just describing. However, this is the link, and this is it's no longer happening. I remember there had been talk of moving all of the data, because there was a couple of these different platforms that we built, moving it all under one umbrella called Tribe. It had some acronym. I don't remember what the acronym stood for. But that, I think it may have been moved over to Tribe, and that's why it no longer works. But here's the actual paper, and let's actually find out what this SVM stands for. Support vector machine. So it's a machine learning algorithm, and when you do run Pilgrim, it runs on a bunch of servers because it's learning the relationship between the genes that you give it. So those are the ones above the red line and the red dots. And then you also actually give it a negative data set as well. So that's one thing that I forgot to mention on this, is that presumably your negative data set, so let's make the negative genes in green. Actually, we'll write it out here, negative. In the ideal world, you'd have your negative in one particular location, like clustered together, and your positive clustered together in a different way, and then it can analyze all the different genes and how they work. Zoned out. No worries, Crystal. Sleep well.
What drink?
Thank you for the hydrate, Crystal. Smikes, you know that GIF that NMD sent us this morning about when that person walks in and everything's on fire? That's probably how my brain works. But yeah, so you have this map of your negative genes, the ones that you know are not learning in memory. So the ones that you would take are like... There are to go! Thank you for looking at to go! So you would just compare. You'd find genes that are not involved in learning and memory, that have been tested before, and it's like, okay, you take out this gene, the animal still remembers just fine. And then you have your ones that you know are involved in learning and memory, and then it tests all the different genes. You can further filter this based on where the cells are located. So in this one, this is the very first version of Pilgrim, but we did an extra adaptation of it. And there is this whole... it's a database called Fly Atlas, and it tells you every single cell inside the body of a fruit fly. And it's like a data set that it measures RNA expression. It's the first of its kind. Now it's quite outdated, but at the time it was pretty big. So then you could see, okay, in brain cells, this gene is heavily turned on. So that will also let you filter your results a little bit further. So if you're interested in the gene, then you can further analyze it. So here, this is like the setup of how Pilgrim would have worked. Again, I will look and see, Gwen, we can check on Tribe. I don't want to make an account, but Tribe used to have these, that you create your data set for your gene standards. So you pick your genes that you're interested in. And usually what's neat is there's a citation associated with it. So when you're running this algorithm, it already pulls together data that you might, like, from publications that you might be interested in. So this gene, HMMR1, here's the associated paper that proves that when mutated, increases the risk of breast cancer. So that could be a positive gene that you do. And so it's neat that when you input your genetic data, it outputs the citation or the reference that actually accounts for what that gene is. And then you run your analysis. And then, let's see, here's the SVM model. So this is the model that we were talking about just now with these dots, right, in the different color dots. And here are the inputs as well. So I think it's pretty straightforward. We have in red breast cancer known positive cells, negative standards, and so you see there's a difference. Most of the blue is on this side, most of the red is on this side. You filter it out, so there's a bunch that don't fit a particular pattern. Those are the black dots, and then boom, you get this list of new gene predictions that might behave the same way as your ones of interest. It might not, but it also might. And so you have to, it's not an end-all be-all. You have to go test these genes, but you go from thousands of gene candidates to maybe 100, like in terms of a list. And then from that, you can look at the genes and say, okay, which one makes sense, right? Maybe it will pull up actin. Actin is a cytoskeleton gene that keeps a cell from falling like onto itself and falling apart. Probably not actin. Actin does everything, like keeping cells alive, right? So not actin. But maybe you find something that regulates cell growth. Here, SMC2 is a chromatin regulator. SMC2 makes sure that the chromatin state is maintained properly in chromosomes pair inside a cell. It's a good hypothesis, and now it's actually known. If you mess up an SMC complex, the chromosomes don't pair appropriately, and then you get cells with multiple chromosomal copies, which actually can lead to cancer. So that's how this kind of listing works. Does that make sense, Chad? Any questions? Is a rainbow factory? It is not, Smikes. If it's a rainbow factory, it is one loony neuron who was wearing glasses and he fell and tripped and broke his glasses, and now he's got buckets of tie-dye paint and he's just splashing them everywhere. Why? He doesn't even know.
It's time to say something nice about yourself.
I have a friend whose name is Andrew CZ, who's built like a Greco-Roman god, and he's extremely handsome, extremely kind, extremely miraculous. Andrew! We love you, Andrew. And we also love NMD. NMD! And we also love Smarks. And of course, Grimly! We just have to be excellent to each other. We don't have me screaming Grimly yet as a sound clip. We'll have to get that in there so I can randomly trigger it. But yeah, so that's the idea of this, and then of course there's other outputs that you can have with this data collection as well. So like how this curve rises and plateaus, that affects it. Hello, Coldburn. Thank you for the stretch. I actually needed today. I held Baby Alona a lot today, and my right shoulder is a little bit nerfed. here again, like, these are just additional metrics of how good your model is. Hold on. Becca! How was that, Becca? Becca. I can also do like a machine gun, Becca. See, it then devolves, Becca, but it's fine. It's totally fine. What is this paper called, Gwendolyn P? Yes, Gwen.
Where's my drink?
What drink?
Thank you for the hydrate, Colburn. Thank you so much. Gwen, there's actually a really nice companion paper. Big Amy! Master Lee is in his house. Gwen, I'll post it on the Discord as well. Guys, if you want to check it out, lovely and amazing Big Amy with a single greatest father on planet Earth. Bit, we love your face to the extreme. I hope you're doing well today. Guys, go check out Big Amy. You're breathtaking, sir. How you doing, Big Amy?
Excellent to each other.
Grimly is excellent. Hi, Archeros. How you doing, Archeros? Just got back from this game about to have 9 months. What are you having, Big Gamey? What are you having? Before you look, what are you eating? What are you eating, Big Gamey? Tell me things. Archeros, how are you doing today? Antonio, game ready? Oh, yeah. Oh, I have to fulfill the prediction, Gwen. I'm so sorry. Gwendolyn P is right. Did Bullen get haircutted today? I totally forgot. The correct answer is no. Not yet, folks. Spaghetti and salad mixed together or independently, Big Gamey? What kind of salad? Caesar salad? Debs and I were talking about how Caesar salad is our favorite salad today. And then spaghetti, do you do meatballs or do you do like crumbles? I prefer crumbles. And in the ideal universe, you have mostly crumbles and a couple of the meatballs that are like slightly sauteed around the edges. Whoa, it's good stuff right there. Also, another prediction, folks. How many topics will we get it done tonight? I am counting this as one topic, even though this was not on the docket, but it doesn't matter because it means that we're having fun and we're chatting about different kinds of science. So that is at least one topic that y'all can predict on tonight and guarantee that we will at least finish one. But y'all can predict with those channel points as well. Independently, just green salad. Today it's crumbles. Enjoy, Bits Gaming! Enjoy your dinner, Bit Gaming. Debs, your salad looked amazing today. I cannot remember the last time I had that good of a salad. Although there were ones that I've had that good. So actually, there's a really cool follow-up, Gwen, to this kind of study. I know we're rabbit holing really hard already, folks, but stay with me on this journey because I promise you it'll make sense eventually. Let's see, Gug. Okay. So all this salad, who would think of the poor plant? I know, Smikes. I worry about the plants. So, Gwen, let's actually see this with a learning and memory phenotype or experiment. Like in a learning and memory paradigm, how does this actually correspond, and how can you use this gene prediction method? So let's go ahead and check this out. Argan Live had salad yesterday with what? What kind of salad did you have yesterday? Again, I love Caesar salad. It's a very basic salad as a Caesar salad. Like, it just has the lettuce and the croutons and, some cheese shredded and the really good sauce. But, like, I had a Cobb salad, actually, with chicken, egg, even had some corn in there and a couple of other things a little while ago. And that, oh, God, Debs, that had a spicy Southwest sauce. Oh, ho, ho. Debs. So good. Egg salad, listen, Smikes, whenever I hear egg salad, I think of SpongeBob. I'm going to tell you straight up. Guys, go check out the lovely Archeros! Woodcrafter extraordinaire, Archeros. Love your face. I'm glad you're hanging in there, my friend. I hope it gets really good. I hope it gets really good today. Romaine, arugula, Caesar croutons, tomatoes, Caesar dressing. Nice. Argan Live, arugula is a fascinating add-on to a salad. I think it's like a really good additional piece to have, but sometimes can really throw off the flavor of the salad if you have too much. If you have too much for it. But it's just as a possibility for it. The G in my name is pronounced like a J. Argen? Is it Argen? A berry sauce. Debs, at our wedding we had a berry sauce. The salad had edible flowers, fruit, nuts on top of like some kind of lettuce, and maybe there was a arugula in there as well, and maybe a little blue cheese crumble. I don't remember exactly, Debs, but that was absolutely delicious. And I'm usually not at all into any kind of salad with, like, fruit, but... Oh, Eugene Argene. Thank you, Argene. Argene! Look at that. I even put in the rolling R in there. I put in the rolling R. That leftover arugula of the other meal. Gotcha, eat it down. Gotcha. Makes total sense. Makes absolute sense. All right, so... Gwen, this will take you through the process of how you use this kind of data set. So, So here, for example, is the list, like how we ran the actual experiment. So we use that Pilgrim analysis the same way you just saw. You put in known genes that are learning and memory involved, and you put in a series of genes that are not. That you know for a fact are not involved in any kind of learning and memory. Okay? And then you get out a list of genes, and you have a gene score and an estimated probability. The probability varies depending on how high it ends up going. This isn't always the most important measurement. It's really a combination of all these. You want this true positive, false negative rate to kind of go up like this. The ways when it's bad is if it just exponentially shoots up versus like this nice concave curve. So there's different ways of interpreting it. But you can see we got a lot of genes. Debs. Listen, you're not the only one, Debs. And then it brings me back to like that really, really awful pickup line that we learned in our youth. Which I will not go into, but you know exactly what I'm talking about, Chad. So here, Gwen, there's actually a couple of unknown genes here. So Gwen, you see there's two CG genes here. Those are unknown. You have no idea what those two do. Anything that is not like some kind of weird combination of letters, if it's a CG followed by numbers, it's unknown. You have no idea what it does. There's not even any predicted thing of what it might actually end up doing. So it could be interesting for that reason, Gwen, or it could be a very frustrating rabbit hole that you run down. So, Gug was the one that we actually picked. It is short for grunge, and it is the mammalian equivalent of the gene atrophin. What that does, it is involved in opening and closing chromatin. So it is involved in chromatin regulation and gene expression. And interestingly enough, there had been a paper that came out 6 months before this one did, looking at the equivalent in mammals, in mice, and identifying that there was an equivalent to this GUG gene. It's a histone deacetylase, so it removes acetyl groups from histones that end up closing DNA, that it affects learning and memory in mice. That was a Nature paper. This is a G3 paper. It is a very, very different tier of publication. That is the benefit of getting something in first. So if you're the first to identify a phenomenon like that, you will get a higher tier publication. If you don't identify something like you identify something really cool, but you're not first, that inherently puts it down, even if it's still the same amount of cool. So the next big thing was this, Gwen, this is one of my favorite little web.
We Gucci.
That was Andrew's. He's right there. We Gucci indeed. I love your face, Andrew. Speaking of which, I forgot to update the sounds, Smikes, for the Moko command. I forgot to do that. I will do it tomorrow, Smikes. I will do it tomorrow. I didn't finish doing stuff tonight until like 7:30, and then I did for work because I'm not doing anything for that tomorrow, and then I was trying to get the new commands in, and it just totally spaced. And I was getting food for Baby Alone. It was a whole thing, Smikes. I apologize, Smikes. Smikes, I understand if you feel a little... like Gwendolyn P. But I hope you can forgive me, sir. I hope you can forgive me. So you can make this web. No, Smikes, you're fine. Hi, Smikes! I was just saying, please don't be mad at me. Please don't be mad at me, Smikes. Please don't be mad at me. So, Gwen, to further keep going on this, Gwendolyn P., what you end up doing is you get this web here. Oh, Smikes, I didn't put in the new Moko sound alert that you did because I just remembered it. I was working until 7:30, and then I was putting in the new redeems, and I was making food for the little one, and it just slipped. But I will get it done tomorrow, Smikes. And I apologize, sir. Well, I care. Anywho, so, alright, so you get this list of genes, Gwen, and then you do an interactive map like this. The ones in red, the red circles are known learning and memory genes. Those are genes that we know for sure have been tested in the fruit fly, and if you remove that gene, that animal does not have correct learning and memory response. The gray ones are genes that were spit out by the software. This one's not interactive, Gwen, but we can make one if you would like. We can make an interactive one. Essentially, in the interactive one, you hover your cursor over a gene and it lights up all the genes that it's connected to in terms of regulation. Hi, Nelf! How you doing, Nelf? Welcome on in, Nelf! Nelf, we're going down a rabbit hole of mathematical modeling and genetic prediction. How we got here, Nelf, is a mystery, but we're here right now. And this, I promise, does link into the next topic in some way. Maybe, possibly, potentially. Sometimes. So Gug, this grunge gene, is actually, I changed it to red right here. And that one's actually interconnected in this entire web. So it's a really good genetic candidate. So you're trying to identify, Nelf, learning and memory genes. And we got a list of potential candidates by running this genetic analysis. And then you put all your candidates into this software and it tells you how they're interconnected. If they're interconnected at all. These blue lines indicate high confidence levels. Meaning that statistically, there's an 85% chance that they are actually interacting with each other. If you can change the threshold, so you can make it less likely to have an interaction or more likely to have an interaction. And that's how those function together. So that could be a map that we could work on in just a moment. A little Epic of Sam, a little bit Epic of Sam. And then actually, what we ended up doing is doing their the behavioral measurements. I apologize for these graphs. I hate these graphs. I've made so much prettier graphs than this afterwards, but this is what was required of me, and this is what I did. So essentially what we're looking at is here are normal flies. In the light gray bar, these animals are the unexposed. So they have a certain behavior. And then the ones that we're testing their memory for are in the dark color up top. And you see they remember for a couple of days. What you find is that the mutants, and this is a heterozygote mutant, which means one copy of the gene is mutated. The other copy is totally normal. And guys, please ask any questions. Please ask any questions if there's anyone that has something to... Please interrupt me, ask. You can't actually make an animal that has both genes mutated because if you do, the animal doesn't survive. It's an essential gene. And what we find is even having one normal copy and one bad copy leads to this animal forgetting quickly. So it still remembers on the first day, but then all of a sudden, oh, it forgot a lot. Nope, it totally forgot by the third day. So it in fact is a gene that is involved in memory retention, not formation. And then here, Gwen, what you can do further is you can turn off the gene specifically in a particular region of the brain. So this OK107 and the MB247, it just is a genetic construct you put into the brain and it shuts off in the mushroom body. That's the learning and memory center of the animal brain. So everywhere else it's on. In the rest of the body, that gene is totally on. Heidi! How you doing, V Game is? Welcome on in, V. V, we're talking about genetics and genetic prediction and all that jazz. What info did they forget exactly? So, Gwen, this particular behavior... So this assay, it's a learning and memory assay. So, Gwen, what you do is you have 2 vials. You have your normal, and then you have your let me change this around. We'll do unexposed and exposed. And so what you... Raiders! Boyd's Custom Fab! Raiders!
Science stream, but we ain't bugging. We over here bubbling. You be chugging what we got. Now the tribes arrive. I promise that I have prescribed you vibes. The vibes we got are giant. Shout out to the stream for the raid. That's science.
Raiders! What are we talking about tonight? What are we talking about tonight? It is Grab Bag Science Night, and we got thrown off on a... Hi, Roland Art. How you doing today, Roland? Welcome on in. We got thrown off on a tangent to start with. And actually, I think it's a pretty fun tangent. Do organisms such as fruit flies with very short lifespans have capacity to create long and short-term memories? Smikes has given us a reset. Thank you, Master Smikes. Okay, great. Let's actually pull up what we were just chatting about earlier. So, the answer is yes. The answer is yes. They can, in fact. Buttsbot, So, the topic even before that, boys, was can we identify new genes and new behaviors for genes that we didn't know of what they did beforehand? So that was the first step. And so what you were able to do is that there is a program called Pilgrim. It's named after the explorers. It's now lumped into a server called Tribe that has multiple genetic predictors. I cannot remember. It was Pilgrim and another one that got molded into Tribe. I don't remember what the original was called. Essentially, boys, what you do... This was the original graph that we did. Someone even say it's logo! Hold on. I always lose this button.
Who let the ants out? Let's go!
Those Moco logos. So, boys, what you do in running this software, it's actually pretty straightforward. You select genes that you know are involved in something that you are interested in. So, in this case, the experiment we're running on this image that I drew out, these red genes, we know are involved in learning and memory. So, you pick a certain number of those. You pick another certain number of ones that you know for a fact, like the ones in green, are not involved in learning and memory. And then you run a machine learning algorithm on it. The machine learns that, okay, all genes that behave like this are involved in learning and memory. The genes that behave like this are not. And then it adds in new genes that were not on the list to begin with. And so the idea is that these blue spotted genes get thrown into it. Hi, Theo!
Back in Gucci.
What is studied to identify the gene behavior? Is that so? It depends, boys. So this data comes from RNA sequencing, which means you basically so you'll do something like this. So you take 2 different types of organisms or a single one. Has yours been? I don't know, Theo. Not a lot of Gucci's tonight, Theo. Not a lot of Gucci's tonight. So, boys, for example, you can take a normal wild type cell. Zach and Rutledge, sir! Zach, thank you for the resub!
It's all Gucci.
You Gucci soul. Zach, how are you doing today, my friend? Welcome on in, folks. If you're not checking out Zach M. Rutledge, Lego builder, amazing voice. Just like Zach.
Gucci.
your voice is very Gucci. Okay? Please go follow Zach. Go lurk on Zach. He's on his partner Poosh. He's on the final stages of the partner Poosh. Go follow him. Go lurk him. Hang out with him. And redeem his hydrate command. He has one of the best hydrates in the business where he makes you feel special. He makes you feel special when you get him to hydrate because he salutes you. I know, Michael Hasser. How you doing, Michael Hasser? You've been nerfed, boys. No. No, Zach, no. Zach, sir.
Science, know you. You're the cutest. I know that you deserve it, Juno. You a star over here. I'll reverse the card like Uno.
You're breathtaking.
No, you, Zach M. Rutledge. You handsome devil. Zach, if you insist, Zach, on being rooters, patooters, and saying lies, that'll make me angry. And then, Zach, after I get angry, I might even cry. But at the end of the day, Zach, I still always love you. Even with egg on my face. Look at that. We tied a bunch of new alerts together. Look at that. Well done, chat. Well done for us. All right, so boys, what you do is you get RNA level sequencing. What that means is you measure the genes and how much RNA there is in different cells. So the example that I gave here, you can yeet a cell into a machine and you can measure this gene called beta-catenin. And you can see how are the levels, how high or low is the mRNA. Remember, you go from DNA to RNA to protein in most cases. So right, if you have a high amount of RNA, usually sometimes that corresponds to a high level of protein. Not always, but sometimes, most of the time. So if you have, high levels of this, that might inform you that it's important in a particular behavior. So if, let's say, boys, we're testing for learning and memory, you expose the animal to a stimulus and it remembers 24 hours later. So maybe at 24 hours, you take its brain and you sequence the information in there. And you find there's a cluster of genes that are really, really, really highly turned on and some that are really, really turned off, that are different from animals that didn't learn a thing. You might predict then that those genes that are different are important in learning and memory. Maybe. It's not confirmed, right? Because all you've done is you've gotten measurements. The only way to confirm that is to actually remove the gene. Like, you go into the animal, you get rid of the gene, and then you say, is it disrupted? Is learning and memory disrupted? By getting rid of that one gene. That's the only way to actually prove it. Right now, you're just proving a correlation. Like, when this animal learns, these genes go up, these genes go down. It's a correlation, not necessarily causation. So, we have the data, boys. We have the data, and let's say we've tested a bunch of genes, and we see that 10 genes are involved in learning and memory, and 10 are not. Great. But, boys, we have thousands of genes we can test. Boys, are you telling me that you want me to spend the next 6 years testing each gene to see if it's involved in learning and memory? Boys, you're not going to pay me that much money. You're not going to pay me for 6 years to test 6,000 genes. You're not going to do it, boys, right? So we need a way to filter that data and make it easier to figure out and see which ones are more important to test than others. Well, how are we going to prioritize that list? Because honestly, boys, when you get that giant list, it's kind of daunting. You have a list of thousands of genes, and any one of those might be the moneymaker that solves Alzheimer's, let's say. I know that's like a very simplistic way of saying it, but in theory, there could be a gene on there that will be the thing that really helps with long-term memory retention. This isn't just memory, right, boys? Like, this could be any phenomena. It could be cancer cell. It could be growth. It could be development. It could be whatever you want. We're just using learning memory as an example. You only cost $300 a month as a student, anyone can afford. That's very true, boys. Boys, listen, boys, as a grad student, I got paid in wacky bucks. Turns out wacky bucks are not legal tender anywhere. But dang it all, boys, I made a whole 5 wacky bucks every month. One time I even got a bonus of a single wacky buck. It was terrible. So, boys, how do we filter down that list, right? So there is this really cool mathematical model that was developed. It's called Pilgrim, named after, like, pioneering into new lands. So, like, discovery of new genes. And it's also an acronym. Oh, gosh, what does the acronym actually stand for? That is... Yeah, hold on. Yes, it is Platform for Interactive Learning by Genomics Results Mining. That's what Pilgrim stands for. What's the conversion rate of wacky bucks to shroot bucks? So 6,000 wacky bucks equals 1 shroot bucks. Thank you, Epico Sam. How do you distinguish genes from others? So Goethe, the way you can identify a gene from a different gene in one of these RNA sequencing experiments is the body of the gene. A lot of these genes are actually identified already in its terms of structure. So if this is a strand of DNA, there is usually a sequence at the beginning of a gene. That's where the transcription factor binds to turn it into mRNA. And so that's a pretty regularly repeating area. So that's when you know the gene body starts. And then, you know, you keep going through the length of the gene. That could go a fair number of distances. It can go a couple of bases. It can go a couple of kilobases, which means a couple of thousand bases, right? And so the lengths vary. But the end site, it almost always ends with a UAG. It's a stop codon. It's a 3 series of nucleotides that means stop. And so your green beginning site and your end red site constitute a gene. Where it gets confusing is that within the gene body there could be introns and exons. So imagine, if you will, you make the mRNA, but there's regions in there that you need to chop out. And so that's just the processing that can happen afterwards. So, like, while this is actually the mRNA, it needs to be further processed, and it's going to be shorter, where it removes those red components. And so there's a whole bunch of other factors in there that regulate the actual genetic information. So even if you have the body of the gene, which this off on the screen, but we were talking about genetics and gene prediction, and Gwen had questions about what the stimulus was for our learning and memory behavior that we're analyzing tonight. So the stimulus, Gwen, and the whole reason we were talking about this was we got a list of genes that we know are involved in learning and memory. Those are our red genes up here at the top, and our green genes down below are the ones that are not involved in learning and memory, but they are just housekeeping genes. Or they're involved in other kinds of behaviors, essentially. So that's a little bit of the background that is happening with what we're talking about. You put it into a machine learning algorithm, and it figures out the patterns. So the green genes are not involved in learning and memory, and it figures out what's unique and similar about them. What makes them different from each other, and what makes them similar. Same thing for the red genes up at the top. What makes them different? What makes them similar? And then it imposes a bunch of genes that were not in the list that you gave it, and tries to place them. So imagine an additional 300 genes, or 400 genes, 600 genes, dots on this graph. And you see where they fall into the cluster. Carlo! Grabs one of his bag. I do, with the one with the fungi. Carlo. How dare. Guys, go check out the stream scribe, aka Carlo. On his partner Poosh. Amazing letter artist is Bob Ross, letter art on the platform. We love Carlo here, and one day I will meet Carlo, and I will give him the most biggest of hugs. And it will bring me so much joy. Octopi, thank you for the follow, and thank you for posting into the Discord. Let me check the art piece. Boo, 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 there it is. Alrighty, let's pull. Oh my goodness, one looks like a yeast cell. I just saw the preview, and I'm like, yeast? I'm sorry, Octopi. I'm geneticist. These are the things that I see. Oh, that is definitely not a yeast cell. But it is quite lovely. Doctor? Coddle. Professor Coddle. Alright, and one more. We got 3 tonight. Heck yeah, Octopi. These are cool. Alright, so these are, folks, from the lovely Octopi who rated on in here. 3 unique animation pieces. Now, Octopi, why did I see this as a yeast cell? So this is actually what budding yeast looks like. It's a big circular body here, and then it has this bud, and that's how they divide, so that's why I'm seeing a yeast cell. Nice! Oh, that's really cool, and I like the oh, nicely done. It definitely looks like an octopus eye as well. Sweet. I like the sounds, too. Nice, Octopi! Number 2. Oh, that's cool. That is really cool. Nicely done. I like that a lot. That is a good loop. Let's check out this one. Oh, cool. I love the loops, Octopi. They're very smooth. The one with the letters going up and down, that on a loop is just absolute gold. I really, really like them. Thank you so much for sharing and for joining the Discord as well. Again, folks, check out our artist friend, the lovely and amazing Octopi. Thank you again, my friend, for the raid and for sharing your artwork with us. Really appreciate you. Again, we're talking about genetics and gene prediction tonight. Again, we rabbit hole on accident onto this topic. Not at all what we were originally planning on doing, but that's good. Trying to make them phone ready, searching for the confidence to upload them all. You got this, Octopi. You got this, my friend. They're really, really good. Just put them on out there, I think. Look, when I started doing art stuff, I was terrified. Because I'm like, someone's gonna, it's like the one clown's gonna come out and be like, it's total garbage. You know what, sir? You have amazing work. And don't let the haters hate. You do good work. Just gotta put it out there. It's hard, but it will make it better, I think. It builds confidence. I would never have had the confidence to do this kind of stuff if I didn't start to put it out there. I know. It's Octopi, listen, it's a little bit like, makes you feel a little bit like this, you know?
Hey, Belen, it's time to say something nice about yourself.
The OMG reaction I made, Smikes, is not that bad. Love you, Smikes. Okay, so thank you for the ants, Smikes. So we're trying to identify new genes. Gwen, if you're still there, Gwen was asking what was the stimulus that we were talking about in the paper we were discussing, how you're trying to identify these new genes. So Gwen, 2 vials, and one of them is your unexposed, and the other is your exposed. The exposed ones is the one that should have a memory response. So that's your tester. So this one right here is the one that's going to show off if there's a memory or not. So, and then this one is the one that we compare to. Now then, the question is there a memory response or not? And what Gwen was wondering, what is the stimulus that we use? So Gwen, let me just double check, because there's a couple of different assays that I've run, and let me make sure I'm telling you the right one. Okay, cool. So this one, you have the fruit flies in your vials. I'm just going to illustrate them with these orange dots. And then in the exposed, we add parasitic wasps. Those are the little black dots. And you cohabitate them for a day. Hello, walking cliche. Welcome in. Talking about learning and memory in fruit flies. So you cohabitate them for a day, Gwen, and then you take the flies out and you put them into a behavioral chamber and ask what food do they lay their eggs on. So here's the chamber, Gwen. It's a petri dish, and there's 2 food options that the animals have. One food option, we'll mark in blue, other one in pink. The pink is 6% ethanol. The blue is 0% ethanol. And then you measure which side they lay their eggs on. Do they lay their eggs on the ethanol food, or do they lay their eggs on the non-alcohol food? Why would they lay their eggs on the alcohol food, Gwen? Alcohol is protected from the parasitic wasps. If the parasitic wasps are there, the flies lay their eggs on the alcohol-laden food because it prevents infection. So essentially, the wasps are not... They are susceptible to the alcohol, and the flies are not susceptible. So the flies will recognize that there's a threat, and in order to prevent themselves from being, their next generation offspring being killed, they protect them by laying on alcohol. Now, the alcohol, why don't they always do that? The reason they don't always do that is because there is developmental delay when laying on ethanol-laden food, and even at higher concentrations, there's mortality associated with it. So there is a trade-off there. How do the flies know about the wasp? So, Smikes, it is an innate search image that they have, so it's all visual-based. So blind flies do not show the behavior. Flies that cannot smell do show the behavior. And you might be Belinda, if they can't smell, how do they detect the alcohol? Alcohol is detected through ionotropic receptors, not olfactory receptors. So there's your answer for that. They have a series of neurons in the lobula, which is in the optic lobe, that if you activate, will do the same behavior. So presumably there's some kind of neuronal signaling from the visual center of the brain into the central body that says, hey, you see a wasp, change your behavior. And Smikes, it is not as simple as that they know that the alcohol is sufficient. What the actual brain does, so there's the eye. We'll make it red, because we have a wild type fly, of course. The fly ends up seeing a wasp. The fly gets scared, and so it has two behavioral options that it can trigger that we know of currently. One is to decrease eggs, so it lays fewer eggs. Two is to lay on ethanol food. So if there is no ethanol food available, it will go down on this path. It will lay fewer eggs until the parasitic threat has left. If there is ethanol food around, it bypasses that whole chamber and it lays on ethanol-laden food. On that front as well, there's an additional option, Smikes, where they will also socially communicate the threat. So regardless of which way it's done, each one of these feeds into social behavior, where they communicate the threat. Does that make sense, Smikes? This is just, yeah, so this is an inherent search image, Smikes, meaning that how we have an innate fear of snakes, it's a very similar message to that as well. So, like, we fear snakes, even though we've never had any prior experience. Same thing happens as well with these animals. They fear the wasps. They have no innate experience with them. In fact, these animals that we tested, Smikes, hadn't seen a wasp because it was the Cantonese strain, which was preserved, like, identified in a lab, or kept in a lab ever since, like, 1915, 1930, between there. So, like, you're talking millions of generations that it has not experienced a predator threat. And yet the behavior is still there. So it's all based on this innate search image that it functions off of. Yep, walking cliche. Most people have an innate fear of snakes. Most people have an innate fear of snakes. Some people don't, but the movement of snakes usually is able to trigger that fear response, which I think is really neat. There is such a thing like that. Treated by the memory part, how will they forget? You got it. Yes, you got it, Gwendolyn P. Gwendolyn's like, Belen, come on. Let's get it together. So, Gwen, let me show you the graph. In pink, Gwen, is going to be the data from exposed animals. And I'm actually going to give you the 10-day graph, Gwen. So this is day 0, and this is day 10. And then this is percent on alcohol of eggs laid. So normal, Gwen, the unexposed is pretty stable, roughly around 30%. Roughly about 30% that you see the amount of eggs laid on alcohol. At day 0, which is when there's still wasps around 95% of the eggs are laid on alcohol if the flies are with the wasps. And then if you remove the wasp, you start to see this decay curve. Hi, Rezzy! How you doing, Rezzy? Oh, come on in, Rezzy. So, Gwen, this decay curve is how there's a memory time where the memory is pretty stable. And then it starts decreasing. In a study I helped with in the Dark Ages, freshman college students in memory experiment tended to remember things at the beginning and the end of the duration of the experiment. Very neat, Nelf. Speaking of snakes, I had a yellow boa snake. He was with his owner on the beach. I was just chilling, held him. It was amazing. Nice, Becca. Becca, look, snakes can be really cute. It's just, I feel like in our adult lives, if we... Like, you would no longer be afraid if you were exposed to it a lot, right, Becca? Like, you'd be desensitized to the fear of snakes. Or if you're an animal, like, you work with them in a zoo. But not everyone can get over that. But so, Becca, like, sometimes childhood curiosity can take over, right? But it's not everyone. It's not like an everyone rule of thumb. But on the rule of thumb in general... same thing with horses, actually, Becca. If you... take like a string or a cat toy and wiggle it around a horse and like shake it around so that it looks like a snake, they'll actually start bucking at it and they'll spook and like run away. And again, that's the same principle. It's like, this horse has never seen a snake before. It's never been bitten by a snake before. Why the heck is it afraid? And again, it's that built-in genetic component. So again, it's not every horse. It's just most horses. They are smikes. They are. Both terrifying. Terrifying one is smart. Yes. I think smikes. I think so. The snake. Oh, yes. Yes. So Gwen is asking, okay, we have this behavior. Why is it, like, what is the cause of this, right, Gwen? You want to know where that memory decay is happening from? So, Gwen, it seemed like when you added in that mutation, that GUG mutation, it's called grunge in flies or atrophin in mammals, what you ended up happening was this. So you had a downshift in the curve. It didn't ablate the memory. It just was involved in the memory retention process, and it seems like it's near the beginning of the memory retention. And the big reason for that it's an important and interesting component, is it is a histone deacetylase, which means it modifies chromatin structure. So most likely chromatin structure is modified to have genes turned on to process and help process memory formation. And if you don't have that gene on that can modify chromatin, you don't have the maintenance of the long-term memory. So you need active changes in the transcription. It makes sense to Gwen that it would be plastic like this, meaning changeable, because if, let's say, the strength of the exposure is very weak, and maybe you don't want to remember for a long time, it makes sense to want to fade your memories faster. And what better way to do it than by regulation of chromatin, where you turn on genes that are important for retention, but if you want to turn it off, then maybe you change it around faster. But with the mutant, it just happened all the faster. The other thing, Gwen, is we have unpublished data on this gene here. So there are ones. So there's 2 genes, Gwen, that can give you this third effect. That third effect is maintenance. Well, so there are many more probably spikes than 2, but there are 2 that I had tested and I had worked with. So what that means is these animals never forget. So Gwen, when you walk into a bar and it's really smoky, would you say that it's stinky, Gwen, or anyone out there? Would you say that it's in fact stinky? If you walk into a smoky bar. For me, it's stinky. And the question then is, Gwen, okay, there you go, Gwen, that's the perfect answer for the first few minutes. Gwen, why do you forget? Why do you forget after the first few minutes? Why does it... Where does it go? Where does that... The smell hasn't left, right, Gwen? Presumably it's still as smelly as it was when you got in there, right? They didn't clear the air because you walked in. So where has it gone? It's gone... The smell hasn't gone nowhere. Instead, you have this thing in your body and in your genetics called memory extinction genes. And they keep you from being hypersensitized. They turn on when you're stimulated like that. And that is, your body forgets about the smell. There are 2 genes that have been identified of this. Their labels are, we're just going to write it up here, DYSB for dysbindin and SYN for synapsin. It seems like these 2 genes, if you get rid of them in the animal, they never forget. It just flatline. When I did the experiment, I took it out for 20 days. It looked the same at day 1 as it did at day 20. They never forgot. Now, that doesn't mean they eventually won't forget, but like, you never saw them actually forget. So it had been and actually we tested in 2 different memory behaviors. We tested in this one, actually 3. We tested in 3 different memory paradigms, Gwen, and got the same answer every single time. And so it's an advantage to forget, Gwen, actually. So if you're like, think about it with this behavior. If you're always laying on alcohol-laden food, and you're preferring higher and higher concentrations, like, you're gonna have infant mortality. That is just something that happens, like with fruit flies. Like, a higher percentage of alcohol, fewer and fewer hatch because there is mortality associated with it, right? If there's no predator threat, then there's no reason not to just lay all your eggs on regular food. And so there is not forgetting is a disadvantage, Gwen. So especially in this case, right? If you never forget the exposure and you always have your behavior on this particular path, that will be a bad long-term thing. Same thing with, like, if you're in that smoky room and you can never turn off your nose. Imagine if, Gwen, inside that bar there is a lion. And you don't smell it when you go in there right away because it's so smoky. But then you get used to the smoke and all of a sudden you smell this other thing, like, oh no, it smells like wet cat. I don't know. In this analogy, there's a pool in the bar and the lion just came out of the pool. Okay? Stay with me, Gwen. Alex the lion indeed. Although, Andrew, that Alex was Alex. So that is actually then, right, then you've gotten rid of the thing that you're used to, which is the smoke, and all of a sudden you can detect other odors. Actually, Gwen, these genes, these memory extinction genes, researchers are trying to figure out how to trigger for particular memories. So, Gwen, what's the advantage of forgetting? Imagine a person who's undergone some kind of trauma, whether it be war or something domestic, right? And wouldn't it be amazing if you could trigger the person forgetting that particular memory? And so by turning on genes at a particular time of like memory trigger and recall, that could help get rid of the whole memory. And that would be a benefit to that individual. So that's one of the reasons why, Gwen, we'd want to look at this. Now, the alternative too, Gwen, is your point of there are advantages as well, right? So, Gwen, let's say if I could nerf my like negative experiences by turning off the extinction genes, right? So you can't do their thing. But then, Gwen, what about the opposite? Gwen, what if I want to know and never forget that you are from Colombia? I keep forgetting, Gwen. I keep forgetting. I want to not forget anymore, Gwen. This is very simplistic, Gwen. Please don't quote me on this. But Gwen, maybe it's super simplistic, but I fire in the neuron that says Colombia. I turn on the memory gene, the extinction gene. Andrew, thank you for the biddies. Andrew, CZ. Love you, Andrew. Love you, Andrew, CZ. So Gwen, I turn on the gene that of the memory extinction. Maybe in that now I remember that very long term. Hi, Bean. Guys, Celestial Bean are in his house. Not only is she a fancy pants with the artist, but also a founder here and a breathtaking artist extraordinaire. Bean, you tricked me today. My mods are much faster than I am. Thank you, mods. Bean tricked me. Bean went live, and then Bean was in someone else's channel. And Bean, you 100% went live today, and then I was like in another channel, and I was like, what the heck is this? Where am I? Where am I? I was scared, Bean. I was scared. Bean, you're breathtaking. How are you doing today, man? I hope you're doing amazing, chat. Bean is an amazing artist, and remember to go buy her coloring book. It is beautiful. It is loving. When my daughter can color, she will be having a Bean coloring book. Confirmed, chat. And she will know what love is. Oh, you hit go live on accident. Bean, I was there. I was so confused. I'm like, is there a collab stream happening? And I'm like, I didn't see you. And then I just clicked the back button, and I'm like, there's still no Bean here. What is happening? I was so excited. You thought you had recorded a bunch of... Look, Bean, on the plus side, it does mean you're breathtaking, that at any moment's notice, we just show up. Okay? So I would take it as a positive and a slightly silly willy. Bean. Slightly silly willy. How you doing, though, Bean? I love your face, Bean. Bean, we're talking about learning and memory and gene prediction, and just like it's Bean, not at all what we're going to talk about tonight. I was going to be talking about nuclear architecture and cancer cells, but you know what? We're having a great time. We're having a great time, Bean. Welcome the heck in, man. Welcome on in. Chat, Bean also has put together the beautiful stage for the SciAnts band. A couple of things. We're still working out the details for making the music video. We're also going to get some... we've got an animated image from Bean, and it's like the background, y'all, it's interactive, like stuff is happening in the background. It's really, really cool. And then we're gonna get a couple of static images as well. And Bean, what I'm thinking would be so cool is to have like an image, like a postcard we can sell, and it says like SciAnts World Tour. I think that would be really, really cool. So like that would be great. Yeah, 100%. Banana. Chiquita Banana. Welcome on in, 100%. Tab here. Yep, for the band, Gwen. I don't want to show you yet what the stage looks like. I do have it, but we're not yet ready to debut it yet. But we'll be getting the static one as well, and the interactive animated. I'm so excited, y'all. Bean is such a talented artist. I love Bean's face so hard. Bean, I love you. Thank you for doing that again, Bean. You're far too sweet. Far too amazing. Love you, Bean. So we were talking about learning and memory paradigms in fruit flies and how to predict new genes and how to identify what the genes did and how they work. Gwen was asking about the stimuli that we use. So Gwen, this is one stimulus. Gwen, this is one. The VIP alerts. Yes, but no pressure. No pressure on Lady Lara Croft. 0% pressure on Lady Lara Croft. So Gwen, this is one of the behaviors that you asked about. So Gwen, to make things a little bit more fun, in the study, there were 3 different ways that we tested learning and memory. So one, Gwen, was the one that you just looked at. The dish. And I'll draw the graph again, just superimpose it here. We'll have regular, and then we'll have the alcohol food. Okay, so that's behavior number 1. There were 2 other behaviors in this, which I think is really cool, that we were able to do in this study to figure out, Gwen, what that GUG gene did. So number 2, Gwen, Number 2, Gwen, 2. We used this apparatus called the fly condo. So Gwen, it was, let me use black here. It was a whole bunch of tubes interconnected, or they're individual tubes, but there were a series of 24 of them. Debs, you're breathtaking. Sleep well. I love your face, Debs. Thank you for being amazing, Debs. What are those things? Oh, it's Patrick and it's SpongeBob at the Fry Cook Games in 2001. My sister got this shirt for me in 2001.
SciAnts know you're the cutest.
Most you, Debs!
Juno, you a star over here. I'll reverse the card like Uno.
Most you, Debs! I will not stand for anything else, Debs! Sleep well, Debs. I watched a parasitic carnivore caterpillar that infiltrates an ant nest pretending to be a queen, eats all larvae, grows like 100 times the original. So, did you, Custom TV, see that on Facebook? I think I saw a similar clip of that. It was like a BBC docu short. I need to dig that up. That would be a fun thing to talk about. Grimly, you saw it too. I need to find the link. And then we can talk about it. I like to talk about it with visuals, and so if we don't have the video, it's going to be so nebulous that it's just going to be like an absolute disaster. Custom, remind me, and I'll dig up the video, and we'll have a chat about, because that's a really, really cool one, and it's a lot of olfactory science in there, which we could talk about. Is it legit? I mean, so it's, Gwen, it's a BBC video, a David Attenborough narrated BBC video. It is legit. Like, it's just a short from the documentary, and they were just putting short clips on there from the BBC's page, BBC Nature's page. So I think it's still okay. But we'll absolutely check it out. I think it's flame on indeed. Yep, mimics the sound. Yep, there's that sound that it does as well. And so that clicking sound, which I think is really cool. It's a unique behavior for sure. And I haven't seen a lot of different ones do that, actually, which is really neat. So Gwen, behavior number 2. This is the fly condo. And we have 2 condos, actually, that we run. Let me draw a second one. And again, you have an exposed one and an unexposed one. Again, you use parasitic wasps. So we'll make... Okay. I'm going to mark them. That's the unexposed again. And that's the exposed. And so in here, there's no alcohol. There's no alcohol in option number 2. Instead, you simply count the number of eggs that they lay. So unexposed, lay a lot of eggs, Gwen. And normally in the graphs, you set the unexposed numbers to 100%. So you graph a percentage instead of the raw number. You can graph the raw number, but then it gets a little bit hairy because they do lay fewer eggs as they age, and then it's a little bit of a murky question, which you'll see why that is in just a moment. Buttspot, you are so behind. So, Gwen, the exposed flies lay fewer eggs every single day. Thank you, Grimly! Grimly, why are you such an amazing legend, Grimly? I love your face. Chat, if you're not checking out Legendary Grimly, please go check out Grimly. Possibly, potentially, maybe, streaming soon. Possibly, potentially, maybe. We love you, Grimly. You are amazing, sir. Grimly, you're... Hold on. Andrew wrote this beautiful command.
Excellent, Chewy Chub.
Immaculate printing, packing, question making. Grimly does it all. The model with the most wisdom and the largest heart. We're so happy that his beautiful soul found its way into our community. 110%. Grimly, I love your face. So, Gwen, the red is fewer eggs laid. Now, Gwen, they will start laying more eggs again. So, Gwen, if we, like, string out the pattern here... That's such a tiny one. Here we go. So it starts off low, but then eventually it will come back up. So that's another... It's the inverse of this. So the graphs are backwards because it's a different thing that you're measuring, but it's the same principle. They change their behavior, and after a certain amount of time, they revert their behavior. The third one... We actually use this different... This is over the course... I only put 4 bar graphs, Gwen, and you'd probe every day, just like in the upper one. But yeah, these are all with 10 days. And you check daily. Bean! Bean, can I show you? We have 2 new animations today, Bean. 2 new animations. You can trigger an angry Antonio now if you type exclamation point rage.
Gucci.
That was Debs. If you type exclamation point rage, you can trigger an angry ant. Bean, like a very if you get, if it's one of those that's science that you're like, you know what, I'm mad about this. I'm mad. I don't like this. Then you can rage. Hello, Moohoodles. How you doing, Moohoodles? Moohoodles, hello, Moohoodles. It is for subs only. It is behind that subs only. But Moohoodles, because you hit that, I'll give you a freebie. Science communicator extraordinaire, Moohoodles, an occasional retro gamer. Guys, go check out Moohoodles. We love you, Moohoodles. We love you, Moo. And the other one being, if you type exclamation point Amber...
Say that.
Gucci and the Hoochie Mama.
It's pretty Gucci animation, I must say. No whoops, Moohoodles. Hi, Moo! Doing well. Tired. Little one, I was with the little one most of the day today. And my arm is kind of sore because we were rocking around and doing a bunch of stuff. But tomorrow is going to be a nice, relaxing day. And so it'll be all good. So, Gwen, the third behavior is probably the most complex, but I'm going to show you, Gwen, what the third behavior is, and then we're going to probe how that one works, like what the paper actually talked about. So, Gwen, the third one is called the fly duplex, and the reason it's called the duplex is because it's just 2 units that are side by side. In one unit, Gwen, you have the wasps. In the other unit, you have the flies. So this is like leaning on Smikes' question, because Smikes was like, okay, if these animals, are they detecting these through visual cues, olfactory cues, or how do they end up doing this detection? And so it turns out it's through visual, because right now, like Gwen, with this particular setup, they can see each other through these two chambers, but they cannot smell each other. And then the chambers are actually plugged. There's actually a cotton ball that we use to plug them, so that the animals can't get out. But for the sake of argument, I'll plug it with the color like that. Super tired also because I didn't sleep last night. Well, Moo, you were live till like 3 or 4 in the morning. Moo Hoodles. Moo Hoodles, there could be a correlation with you being a legendary late night streamer, Moo Hoodles, and telling us about the science of space. Moo Hoodles, you think I don't know, but I do in fact know, Moo Hoodles, because I do in fact lurk on you, ma'am. Constantly tabbed up, Moo Hoodles. Early? Yeah. No, Moo. Maybe early in your time zone, but it was late for me, and I was like, why am I up? And then I was up with Carlo the stream scribe until 4 in the morning. That's not my fault. That's Carlo's fault. He's very engaging and beautiful and handsome, and he's distracting. What are you going to do? So, Gwen, this one is a similar behavior. 2 a.m. Moo, listen. Always have your lurk. We got to do that partner poosh, Moo Hoodles. We got to do the partner poosh. Gwen, this graph, it looks very similar, because here you also do the egg counts, but it gets more complicated. So what you actually do, Gwen, is this is day 0. Day 1 is a totally different experiment where you again have a new duplex, and you have your original flies, Gwen, that saw the wasps. So these flies are experienced, they have the memory, and we call them teachers. And then, Gwen, in the neighboring compartment, you don't add wasps this time. You add new, naive students. And the question is, Gwen, can the teachers confer the information of a predator threat to these naive students? And if so, this would suggest that it's visual. Right? And so the graph, we're just going to graph, it's actually 3 different bar graphs, because there's also the unexposed. Remember, there's the blank that's never seen anything before. So the unexposed, I'll do in blue. You always see the unexposed laying tons of eggs. Right? Your exposed, which is these little blue flies, lay fewer eggs. Right? And Gwen, that makes sense, because we exposed them to the wasps, they saw the wasp, they lay fewer eggs, just like the above graph that we talked about. So your question then is, what do the naive flies do, the students? It turns out they also lay fewer eggs. So even though they themselves have never before seen a wasp, they've only seen your girl! Chat, if you're not checking out the breathtaking, amazing Yoga Garrel, please go follow Yoga Garrel. Yoga Garrel is supermodel extraordinaire across the country of Canada, professional Tetris 99 player, and so much more. We love Yoga Garrel here, folks. Please go check out the amazing, the breathtaking Yoga Garrel. In fact, she is so amazing that she has her very own emotes.
Curiouser and curiouser.
Right there, and the message is pinned to the top of the chat, so you can look and see the beautiful Yoga Garrel for the next 20 minutes. For the next 20 minutes, you can check out the best emote right there, and she's almost at 300 followers, folks. She is... At last check, she was 11 follows away from 300. Chat! Very, very close to that 300 follower mark. Please go check out and follow Yoga Garrel and get her over to 300 followers. Aye, chat! You will not be disappointed. You will never, ever, ever be disappointed when it comes to Yogurt Garrel. We love you, Yogurt. You lovely stream tonight, ma'am. Lovely stream, as always. So, chat. Next question. For Gwendolyn P. Gwen, how does this correlate back to that gene that we identified? So, Yogurt, we're talking about gene identification. Oh my goodness. No smikes. About genetic identification and new genes that are identifying this. No smikes. I win. I win! Oh. Now. No, you can't. If I've nerfed the comments, Mike, there's nothing to pin. Oh. We can actually pin that one. Here, look at that. Boom, look at that. It's a love for Smikes. It's a very good pin. It's a very, very good pin. So, Gwen, what happens if you introduce that mutation? If you introduce that GUG mutation, what happens in each of these memory paradigms? We talked about it was for learning and memory retention, right? So, in the ethanol experiment, which we're measuring how long they lay on ethanol, we're going to do the mutant in dark blue. It ended up being something like this. So they forgot faster. In this behavior with the egg number, they forgot faster. And in this experiment where they were supposed to, we tested their ability to be students. So did they have the ability to receive information and interpret it from the teachers? You actually get this intermediate phenotype at first, so it signifies that yes, they're able to at least in part get that information, but then they don't at all. So it messes up each one of these phenotypes, and that's how like this GUG or grunge gene was so involved in their behavior. Does that make sense, Gwendolyn P? As what the stimuli were, and then we'll get back to what the paper was about, if y'all would like. If y'all would like. But absolutely no pressure to. We don't have to, of course. E? Okay. CelestialBean.com? Sure, Smikes. We can pin that. No problem. No problem at all, Smikes. Okay, so the paper... So these then, Gwen, I think maybe the graphs now make more sense. These, right, are where you see a faster decay of memory. And these are when that GUG gene is only turned off in the learning and memory center of the insect brain. So this previous graph here had it off in most of the body of the animal. And here it's just off in some of the key neurons. You see it's a similar result, so you know that your original result wasn't because the gene was turned off in the leg, but rather it was turned off in other body parts, which I think is really a unique feature of it. And then, here's the other kind of behavior, Gwen. I like these graphs much better for some reason. I was allowed to have my prettier graphs on this particular one versus the other. Gwen, I don't know. I don't know, Gwen. But here, so your wild type flies with the egg number. So the blue is the unexposed, red is exposed. You see the red lay fewer eggs over time. Because they're laying fewer eggs, that's showing a memory. But here are these GUG mutants again. You see, they do remember on the first day, but then they quickly forget. And then here are... These are their teaching abilities as well. So this one actually just reversed that final experiment that we were talking about. This is using them as teachers. Hi, Hugh! Welcome in, Hugh! This is using them as teachers, meaning that when they're exposed to the wasp, they forget, or they remember the exposure. They're able to transfer that information to naive animals. So you see both pink bars are low, but on the second day, both pink bars start going up. And so they lose that they forget that they're communicating a threat because they've forgotten about the threat as well. And the model for this is that this is the idea that we had. See, what happens if you expose them again? Oh, Gwen, that is a very good question. So repeated exposure to a normal fly, it elicits usually a stronger response. For other behaviors that are learning and memory based, they tend to become faster. We never worked out the temporal mechanics very finely. We did know, Gwen, that 24 hours is usually what we did for exposures. We did find that I think it was 7 hours was not sufficient and 14 hours was sufficient to trigger the memory. So, Gwen, you could do a series of experiments where, like, we never ended up doing it because it wasn't, like, it wasn't a priority at the time to figure out the dynamics of it, but you can see if 2 7-hour exposures are the same as a 1 14-hour. You would still have to figure out how much time, like, how much of a gap that you put in between those two learning and memory paradigms, right? Like, if you give the animals 7 hours on, 7 hours off, and 7 hours back on, is that enough? Or does it have to be 7 hours, half an hour break, another 7 hours? Like, those dynamics are hard to know. Usually, though, in other behaviors, like the associative memory, where you pair an odor with a stimulus, so like an electric shock with an odor, then you'll, if you keep doing it, it'll be faster. And it will last longer as well. But it's not clear for this behavioral paradigm yet, just because it just hadn't been worked out. And so the behavior, like we thought for this, Gwen, is that there's an initial stimulus. It then triggers neuronal activity, and we talked about that learning and memory center, the mushroom body. Then it activates early memory genes, and it yields to memory formation. And then we thought there must be a secondary pathway, and this is where your GUG is. So that's an histone deacetylase. It removes acetyl mark. So if when you form a memory, you put these acetylation marks on histones, it turns on gene expression of some genes. Right along here. Spectrotronic grow mushrooms, very nice. Spectrophonic. You turn on these genes. If you want to sustain the memory, you turn those genes off. And that's what you use your GUG for. And presumably, if you cannot turn those genes back off, you don't get the memory retention. And so there's different genes involved in different things. Our guess, Gwen, is, remember we talked about there's those genes that are involved in memory extinction. If those don't get turned on or off appropriately, you won't get long-term memories formed. Because sometimes, Gwen, it makes sense that you don't want a long-term memory. You only want a short-term memory because of what kind of stimulus it is. Does that make sense, Gwen? And so that's kind of the feeling on this. I think, chat, I think we have some videos on learning and memory in insects that might dovetail well on this topic. So, Gwen, there is the goal of being able to delete memories. There's this another DNA modifier called ACSS2, and that has been shown to wipe out certain memories, in particular in mice models. So if you have a mouse and you do an electric shock associated memory, let's actually find the video on that. Where is this? Hold on. Let's watch this one. This isn't on ACSS2, but it's still a good one to check out. By the way, folks, a couple more shout-outs. We also have the lovely and amazing Ristorak in his house. If y'all aren't already following the lovely Risto, please go check out Ristorak88. Twin brother to the lovely Skip, a.k.a. Skip the Bell. We love Risto here to the extreme, so make sure to check him out. He also, Risto got a lovely promotion. A new promotion where they hopefully will appreciate him a whole lot more. Love you, Risto Rock. Love you. All right. Let's check out a little bit on learning and memory. Hi, Smorf! How you doing, Smorf? Welcome on in, Smorfosaurus. How you doing, Smorf? Talking about learning and memory tonight and genetic identification. Not at all what I was originally going to talk about. I was looking forward to... we had a question, Smorf, so we've been rabbit-holing hard into this. Someone asked about, like, how mathematics can interact with, like, biological genetics. And so then we just, like, defaulted and jumped into this whole stream of, like, doing... genetic prediction and mathematical modeling, and then how you ascertain learning and memory in insects, and like how to run the genetic test. And then we went through one of my papers that did this, and so it's been like it's been derailing Smorf, but it's still like a really important scientific topic, and it's a lot. I think we're having fun chatting about it, maybe, possibly, I don't know. I think so. So that's at least been kind of cool that we've been chatting a fair bit about it. But Smorf, I hope you're doing well. Smorf, we're on a cool down on the shout-outs, but I'll give you your fancy pants shout-out in just one moment. Remember, chat, Smorfosaurus, legendary artist, singer, songwriter extraordinaire, and occasional gamer too, and Perler Beater. Perler Beater. That's, Smorf, that's a hard word to say, but I'm working on it, Smorfosaurus, so that I don't know, like my tongue doesn't get tied up when I say Perler Beating. I can't say it fast yet, Smorf. Just like I can't say certain cities and towns in Massachusetts quickly, because I end up eating my words, and that's less than ideal. 4 seconds. All right, hold on. See, Smorf, I can build it up well enough. 3, 2, and 1. Spectra, listen. It was an intentional decision. It was an intentional decision because they just wanted to make sure to not have that reference. I don't know what to tell you, Spectra. Must have been intentional.
Think back to a really vivid memory. Got it? Okay, now try to remember what you had for lunch 3 weeks ago. That second memory probably isn't as strong. But why not? Why do we remember some things and not others? And why do memories eventually fade? Let's look at how memories form in the first place. When you experience something, like dialing a phone number, the experience is converted into a pulse of electrical energy that zips along a network of neurons. Information first lands in short-term memory, where it's available from anywhere from a few seconds to a couple of minutes.
And what's cool is there are genes involved in short-term, medium-term, and long-term memories, and they are different based on like the behavior that you're actually studying. Okay, Becca, listen. It's, okay, hold on. I'm gonna go slow. It's Marlboro. Because when I say it regular speed, I slur it all. It becomes Marlboro. And Lita just chuckles at me because I can't say it the way she does. Because I just eat half the words. Because in the South, that's like a cigarette, or that's like the Marlboro. Right? You got like, if in Georgia, I'd be like, that they're Marlboro. Right? But when I look at that word, it's just, it looks like Marlboro or something, but Marlboro. It Spectra, I think it does, but whenever anyone there pronounced it, it did not have a draw on there. It's how we say it in America. See, Lady Lara Croft knows how it is. Marlboro. Marlboro. I look, because Lady Croft, I grew up in Georgia, so I roll it 100%. And then Lita's driving around and I'm like, what's this here, Marlboro? And she's like, no, it's not Marlboro. It's this, and it's this whole series. Becca, I'm sure there's. I did learn, Becca, Worcester, not Worcester. I did learn that pretty quickly. Worcester, I still have a thing that it should be Worcester, but what, I can't argue with that one, right? The thing with Patois, Worcester, no, Smikes, Worcester. We're not throwing on the extra accent here. Neither are we. We're also not saying rough. It's roof. It's roof. There's an OOF. Also not ka. There's no H in there. There's no H in ka. It's car. It's car. Okay. We're not a bird. It's not H2O Wooder. Exactly, Spectra. You got it, Spectra. Exactly. It's hilarious. Lita apparently used to have a really thick Boston accent when she was little, and she got sent to speech therapy, and so now she doesn't. However, Becca, she can turn it on at a moment's notice. And she one time she turned it on, and she's like, "What do you think?" And I was like, "If you would have had that accent when we started dating, we never would have dated." Like, it just drives me up the wall. It drives me so far up the wall. Oh, and then Lita's like, "That's rude." I'm like, "Honey, you asked what would have happened. Like, I would have stayed very far away from you." I love you, Smikes. You have a beautiful accent, sir. You do not have... No. It's very... Smikes, listen, you do not have the accent. Don't you dare say that. In fact, I'm going to nerf that comment. I love your face so hard. I love your face so hard, Smikes. I love your face, Smikes. That was the most fun and wholesome laugh I had today. Excellent, Spectre. I'm glad. I was from... Sal as well, but I picked it... Oh, Becca, look, I've probably picked up loosey-goosey things as well. You can turn it on, Smikes? Okay, gotcha. Same with Lita. She can turn it on. Need to hear... Gwen, it is... I think, Gwen, the thing is because I'm not expecting it, and she just doesn't sound like that in our day-to-day lives, and so when it's turned on, it's like, that's not you? What is this accent? Put that away. Bring back my wife. Elementary, when I moved to South Carolina to California, they made me say water in front of the whole class. They found it fascinating I said water. Later on, I see nothing wrong with that. Andrew, Hi, Coffee Rocks! Thank you, Coffee, for pulling us back to reality. We're talking about Lita talking in her Bostonian accent. And yeah, it's a whole thing. You probably... Look, Smikes, listen. I can 100% turn on my southern...
Look at its little feetsies! Look at its little feetsies!
That's not... How do you say Gucci? Gucci. There is, Kai, a Gucci that I said in the Gucci command.
Gucci!
That was the ferret, Kai. That was the ferret. That was not me. That was the ferret saying Gucci. I don't even know what to do with that one. No, that was the... Andrew, no, that was the ferret. Andrew, how dare you say that I sound like that? You know I don't sound like that, Andrew. That was clearly the ferret, who's a different person entirely. He's a ferret. The monsters who call it puppet. Now, Andrew and Smikes, I can turn on the southern.
Gucci.
I can turn on the southernism. Smikes, listen, you know you wanted the ferret to have one. I can only hope... No! Andrew, listen, you stop that. You have a beautiful voice. Okay? You're from the land where I like to vacation. I will not take that as anything but breathtaking. Okay, Andrew? Gucci. I don't know who that one was. I don't know who that one was. Either way. Guys, someone else to go follow. Before we continue, make sure to follow our friend Irish John Games. Game dev extraordinaire. Go follow the wizard. That is Jern. Go follow Jern, chat! Where's the link for the nerf? The ferrets there bend your cats. Spectre, we do have the... Let me show you the ferret animation we have. Wholesome ass ferret, aka wholesome as AF.
Gucci.
Listen, Andrew, Lita and I love to vacation in Cincinnati. Okay? Actually, Andrew, before we get back to the video, listen, I had a discussion today with our friend Plain Old Trey. And someone said something negative about Ohio in his chat. And he was like, why do people saying like they're not good at, like, about Ohio? He's like, I love people from Ohio. And everyone just, like, half the chat went off about negative things about Ohio. And of course I'm like, I like to vacation in Ohio. and everyone's like, what is wrong with you? I'm like, I love Cincinnati. Is it Bearcat? Heck yeah, Spectra. Dayton is where we stay, the hotel that we stay. And then we drive over to Cincinnati, which is quite, and that's where we vacation. I love Cincinnati. Hi, Nerdowino!
Ah, you are wholesome as ferret. Yay!
Very true, Gwendolyn people.
Science, know you. You're the cutest. To know that you deserve it, Gino. You a star over here. I will reverse the card like Uno.
You're breathtaking.
Nerdowino, that's inappropriate, Nerdowino, because Nerdowino, the thing is, you're a NASA space scientist extraordinaire. NASA is in his house. Nerdowino has a lovely daughter. Nerdowino, Mini Nerdowino's shirt is now on its way. So it should be there. I think the estimated shipping is late next week. There was a delay on the product, but it should be here. It should arrive to you soon for Mini Guido, who won the T-shirt for the Science Art Gala. And guys, go check out Nerdowino. Not only lovely daughter, but also NASA Space Scientist Extraordinaire. We love you, Nerdowino. We love your face, Nerdowino, you amazing soul, you. Nerdowino. Alrighty. Nerdowino, always, my friend. If I ever don't do it, it's because I spaced out, which is accidentally does happen sometimes. I get overwhelmed and my brain just starts running around and it just misses that I didn't get something done. And that's never on purpose, Nerdowino. I never avoid shouting out our friendos, and sometimes I just get over. I know, and then I make Gwen mad. And that's, I tell you,
Gucci.
Not Gucci. Winslow, you got it. Winslow the Aardvark for you, Becca. Here's Winslow the Aardvark. He's a very cute aardvark. Kevin is fine. I think he's married. So Andrew, just, you know, we got to be respectful and make sure that he knows. Or you mean fine like he's okay? Either way, Andrew is all right. Now I don't judge here. I know. Andrew, I know. I just... I'm sorry, Andrew. It makes me chuckle. It's like, Kevin is fine. And it's, oh my. He's an aspergillus, sir. He's an aspergillus. I know, Chad. I know. I know. I'm very misbehaved. All right. Memories form and how we lose them. Connecting back to our original discussion tonight. Nerdowino, we were talking about genetic ways of genetic tricks to identify new genes, and particularly learning a memory. So we went through gene prediction software, how to run it. We also looked at interactomes of how the genes interact in a network. And actually, that's one thing, Gwen, I did forget to show you. Gwen, this is a really fun tool to play with. And yes, Blunt has weird definitions of fun. Gwen, please don't judge. But I think this is a lot of fun to see this. So, Gwen, let's actually, before we continue the video, we'll go here. If this is a free-to-use platform, it's really fun if you're interested in genes and how they interact. So basically, let's go into fruit flies, analyze the gene. Let's analyze the gene GUG. Rage video. That's the gene that we identified. Thank you, Spectra. We have fun here. So that's the one that we knew that was the new one that we identified in learning and memory. And we can, this is like the genetic network that we were talking about.
I've always wanted to say that.
It's alive! Thank you, Gwendolyn P. So, Gwen, here are a bunch of genes that are interacting, and here's Gug, and so you can highlight Gug and see which other genes in this network is it interacting with. There are a bunch more that we can add, so we can actually make a gene set. Oh, look, we already made a gene set. Look at that. It remembered the gene set. And that's what you can actually do is we save the gene set locally, analyze the gene set. And so now we gave it a bunch of learning and memory genes and the gene Gug, which is that new one that we identified. And so we want to try to figure out, okay, what does that genetic network look like? And so right now it's running, and we get this network. So Gug is right here, Gwen. That's this one right here, and you see it's that big red circle. When I highlight it, boop, it shows every place that interacts. Hi, Twitch Addict! How you doing, Twitch Addict? Good news. Soon I'll be off every other Friday, and then the opposite weekends. Let's go, Twitch Addict! Twitch Addict, that deserves some hype. And no, sorry! I hope you're doing well, Twitch Addict. Welcome to Hack In. So yeah, this tool I think is really neat, Gwen. It's a lot of fun to look at this and check this out. So you can see again, the Gug gene is interacting with a bunch of different ones, and right now our confidence interval is at 85%, which is pretty high. So that's meaning that there's a lot of different, the likelihood of interaction in this network is very high. That it's not accidental that we're getting this interactive map. We also have the number of genes set to 25, which is visually easy to decipher. We can go up higher. So Gwen, this is 75 genes, all with high confidence relationships, meaning that the likelihood that these genes interact is really, really high. What's cool is I can move around the graph as well. I highlight Gug. It links to a great number of those genes. So, Nerdarino, it can be accidental. If we lower the confidence interval, meaning that the likelihood of them interacting becomes much lower. Gwen, look at that map now. This is what Smike's inside look like, apparently. It's a giant spider web. But so, Gwen, here, there's all these greens. So it's actually, there's a... here we go. Graphical options. Low confidence interval is green. Medium is yellow. High is red. And there's... I added the legend back. So legend is on here. So Gwen, now if I look at Gug, it's hitting everything. But because it's also showing the low confidence intervals, it's less likely to be real, right? So like the way that you can actually click on these lines and figure out where this data comes from. So this line that interacts with orb and period gene is what highlights and does circadian rhythm. Orb is learning and memory exclusively and some development. If you click on that line, it tells you that the reason that this is known is because of mRNA processing, that it's a known relationship. It has another known relationship through experimental data through RNA polyadenylation. And then it has other predictions of how they might interact. So these predictions are less likely. They're not necessarily experimental data. It's through like mathematical models that you suggest there might be some kind of interaction that happens. Versus like, let's look at the green one. So then the gene GL, which is, it's a CG identified gene. It's also called GLASS. The GLASS gene, its likelihood that it interacts with the period gene is green. And so it's a very, very low phenotype share. So it says the likelihood that the two different effects are from the same mutant, it's a 0.27 confidence interval, which is really low. Doesn't mean it's impossible, Gwen. It just means the prediction is very low on that front. So that's how it works. Does that make sense, Gwendolyn P? So again, if we wanted to do this low confidence interval, what I would do is lower the number of genes. And see it.
That's pretty Gucci, eh?
That was your girl. It's much more manageable this way. So like I changed it to only 10 genes, and we're also including the weak interactions. So like period and this KIS are still showing, but it's like not especially like great one. Eyes are heavy. No. Good night. Sleep well, Spectra. Sleep well, and thank you for hanging with us. I appreciate the heck out of you.
Hello there.
Hi, Tactics.
Hello there. Hello there.
How you doing, Tactics? Welcome on in. But yeah, so Gwen, if you wanted to do this like for reals, you know, at least the suggested one is probably 0.7 to start with, and then you can turn up the number of genes. Even with that, you start getting this giant web, and what's cool is you can actually click and drag these nodes and pull them away, and so you can see how they interact. And when you highlight them, our GUG gene, see, look at all those interactions that it has in this map. I will add the link, Gwen, into the mention on Discord if you would like. It's a really neat feature, and again, you can click on these lines and it'll tell you what the experimental evidence is as well. Like here, shared phenotypes from memory genes. And also what's cool is if you scroll down, Gwen, it'll tell you your biological processes in your gene set, what is it enriched for, meaning all your genes that you put in, what is it most interacting with? And so you can see associative learning, olfactory learning, and then it gets into developmental cell growth, regulation of organ growth, stem cell development, short-term memory, long-term memory, neuronal signaling, synaptic signaling. So it's a fair number of things that are being pulled together based. When you came in, we were talking about genetic networks. This network in particular is one of an interactome of learning and memory genes. And this is what Gwen was asking about, is if you highlight one of these genes, how will it interact with the rest of the map? And in particular, we were looking at this gene called GUG, which is grunge, which is equivalent in mammals of atrophin, is it a histone modifier. And so if you click on that gene, you see that all these others very much light up. And this is unknown to be a learning and memory gene, but when you click on it, it interacts with this big learning and memory gene here, this other one here, and then this series as well. When you click on these in the series that it interacts with, it hits a lot of the other ones too. So we figured out, okay, this is the interactome between these. And this is a really interesting map that you can do. And what's cool is, I think it's loading. This is a new feature, Gwen. I haven't seen this one before, but it's what it's supposed to do is when you run the new analysis, you can actually see across multiple species how this genomic network interacts. So you can see how it happens in nematodes or C. elegans, and how it happens in zebrafish, aka Danio, which is the organism. I think we're having a loading issue right now, but that's what you should have multiple graphs that pop up. And then this process network tells you what is the most represented process in this genetic network that you made. And you would predict that it would be learning and memory because that's what we kind of pushed it towards. And so you can see this whole one is like light stimulus regulation, transmembrane transport, synapse structure, sensory perception, transport, thermotaxis, establishment of memory, things like that. Yeah, it's a really neat tool, right? Hi, Clement! How you doing, Clement? Welcome on in, Clement. We're just talking about learning and memory and genomic prediction. We were finishing up our video on how memories are formed and how we lose them. Let's go back to that video. And then I think I have one final video to round out the evening, which I think would be lovely to have.
It's then transferred to long-term memory.
So this is where, just to reset, we were talking about how memories are formed, short-term, long-term, medium-term memories. And the fact that there's these different levels of memory, and all about like, there's different genes and different amounts of signaling that'll contribute to if you have a short-term memory on something, if something's a medium-term memory, or if something's long-term memory, and how it's maintained and processed in the brain is dependent on very many different factors, which I think is really cool. When I had a deja vu video so I can show it to people. Like Dame Karen, you mean like a good video on a deja vu phenomena? I can dig and see if we can find something. That might be an interesting and fun topic to talk about anyway one night.
Through areas such as the hippocampus and
hippocampus is the mammalian learning and memory center. It's the same as the mushroom body in insects.
Finally, to several.
We were talking about, again, learning and memory raiders, and we have a video that we're going. Valentine, thank you again for the raid. Appreciate the heck out of you, my friend. Let's go ahead and continue on this view.
Neurons throughout the brain communicate at dedicated sites called synapses using specialized neurotransmitters. If two neurons communicate repeatedly, a remarkable thing happens. The efficiency of communication between them increases. This process, called long-term potentiation, is considered to be a mechanism by which memories are stored long-term. But how do some memories get lost? Age is one factor. As we get older, synapses begin to falter and weaken, affecting how easily we can retrieve memories. Scientists have several theories about what's behind this deterioration. From actual brain shrinkage, the hippocampus loses 5% of its neurons every decade.
And that is not where hippos go to school. But I like that a lot, Smikes. I did see that, Smikes.
For a total loss of 20% by the time you're 80 years old. To the drop in the production of neurotransmitters like acetylcholine, which is vital to learning and memory. These changes seem to affect how people retrieve stored information. Age also affects our memory-making abilities. Memories are encoded most strongly when we're paying attention, when we're deeply engaged, and when information is meaningful to us. Mental and physical health problems, which tend to increase as we age, interfere with our ability to pay attention and thus act as memory thieves. Another leading cause of memory problems is chronic stress. When we're constantly overloaded with work and personal responsibilities, our bodies are on hyper alert. This response has evolved from the physiological mechanism designed to make sure we can survive any crisis. Stress chemicals help mobilize energy and increase alertness. However, with chronic stress, our bodies become flooded with these chemicals, resulting in a loss of brain cells and an inability to form new ones, which affects our ability to retain new information. Depression is another culprit. People who are depressed are 40% more likely to develop memory problems. Low levels of serotonin, a neurotransmitter connected to arousal, may make depressed individuals less attentive to new information. Dwelling on sad events in the past, another symptom of depression, makes it difficult to pay attention to the present, affecting the ability to store short-term memories. Isolation, which is tied to depression, is another memory thief. A study by the Harvard School of Public Health found that older people with high levels of social integration had a slower rate of memory decline over a 6-year period.
And this actually, there was a paper that just came out that we have been wanting to talk about for Science News Friday, is actually looking at how social setting affects learning and memory and aging. And so it actually, if you don't have a lot of interactions, like social interactions, even not even as you age, but like in everyday life, it actually increases memory loss and decay, which is quite fascinating that there's a social component to maintenance of your like your neurons and your like even your brain anatomy. Like your neurons actually will become multinucleated, which is a sign of accelerated aging in the brain. I know, Dame Karen. I know. I'm just telling you what the data says, Dame Karen.
The exact reason remains unclear, but experts suspect that social interaction gives our brain a mental workout. Just like muscle strength, we have to use our brain or risk losing it. But don't despair. There are several steps you can take to aid your brain in preserving your memories. Make sure you keep physically active. Increased blood flow to the brain is helpful. And eat well. Your brain needs all the right nutrients to keep functioning correctly. And finally, give your brain a workout. Exposing your brain to challenges like learning a new language, is one of the best defenses for keeping your memories intact.
And also, Valiant Cheese, you're a great photographer. It's all bad news, but Dame Karen, we're going to finish off with some good news. I can't have it end on bad news, Dame Karen. Dame Karen, there are things in that video that we're worried about. The science we're worried about. We're worried about our brains. We're worried about a lot of different factors. However, Dame Karen, what if we can utilize insects and their brains to ask how we can correct all those negative things that we just saw in humans? Because insects like fruit flies, bees, ants are a model organism for something more complicated. So we can model a disease state in something more simple, like an insect, and then transfer that knowledge that we learned in the insect into a human. How to maintain your mind? Oh, thank you so much. Thank you so much. No, Dragnrock, we're not playing that. But thank you. It's because cooking a priority when you're... Clement, it is not. It is 100% not. It is not on my front either. But yeah. It's, let's make it better, Clement. Let's make it better. Let's talk about how we can utilize the insect brain to learn about factors that would otherwise not be able to be known. Exactly. Save me, Drosophila. Dame Karen's kicking it off for us. And the big thing is, Dame Karen, the reason I think this video will be a positive spin out at the end is because there are a lot of... there were scientists who worked on mammals that when I was giving research presentations in undergrad and talking about the insect brain, their first question was, flies have brains? And this is, again, a scientist, they're working on mammals, but they're still, you would say, PhD within science, maybe knows what's happening on that front. But, no. So that's why, let's talk about why the insect brain is beautiful and why we can utilize it to learn.
The human brain is one of the most sophisticated organs in the world. A supercomputer made of billions of neurons that processes and controls all of our senses, thoughts, and actions. But there was something Charles Darwin found even more impressive.
There's even a book about that, Dame Karen. I think, actually, that's the title of the book. It's called Fly, the Unsung Hero of Genetics, I think. Sleep well, Moo Hoodles! Moo, sleep well. Thank you for hanging out with us, Moo. Thank you for hanging out with us, Moo. And Moo, thank you for not using the very, very scary emoji that you have. Thank you, Moo. I appreciate you, Moo Hoodles.
Brain of an ant, which he called one of the most marvelous atoms of matter in the world. If you find it hard to believe that something so tiny could have a complex brain, you're not alone. In his project to classify and describe all living things, Swedish naturalist Carl Linnaeus assumed insects had no brains at all. He was wrong, but understandably so. Insect brains are not only minuscule, but in many respects, they function differently than our own. One of the most noticeable differences is that an insect that loses its head can still walk, scratch itself, breathe, and even fly. This is because while our nervous system works like a monarchy, with the brain calling the shots, the insect nervous system works more like a decentralized federation.
Oh, Moo! How dare Moo! How dare Moo Hoodles! How dare Moo! Moo, how dare! Dame Karen, it's that one because that one reminds me of that first episode of Doctor Who, where it's that spray, that the thin membrane skin creature that gets sprayed. I forget what the line is, but spray me, and it's just it's terrifying. And that's what that emoji reminds me of, is that scary episode of Doctor Who. That's why. It moisturized me. Thank you,
one is not
Skip!
tethered by earthly limitations.
Sleep well, Skip 24.
What does that mean? Come.
Sleep well, Skip. Thank you for hanging out with us. Please give a big hug to Callie. And thank you, Andrew. Thank you, Andrew. So the I know, I didn't care. It's terrifying. So the decentralized nervous system, well, that's manifests as if you remove the head of the insect. then they'll still function and live for another 24 hours versus if you remove our head, we're gone. But it still has a huge amount of processing ability, and that's why it's important. It's like my overly attached girlfriend smile emote. Oh, I know that GIF. Or not GIF, what is it? The meme. I know that meme. It's hilarious. Good news praying man's, yeah, but Valiant Chi, they don't live that long without a head. But you're right.
Insect activities, like walking or breathing, are coordinated by clusters of neurons, also known as ganglia, along their bodies. Together with the brain, these local ganglia form the insect nervous system. While an insect can do a lot with just its local ganglia, the brain is still crucial for its survival. An insect's brain lets it perceive the world through sight and smell. It also chooses suitable mates.
Yeah, Dame Karen, they'll actually, if you remove their head, like, so we actually do that experiment with fruit flies.
In dreams,
Zedman!
one is not tethered by earthly limitations.
What does that mean?
Come.
Looking cool, Zedman. Sleep well. Thank you for hanging with us, Looking Cool Zedman. Know that you're breathtaking, know you're handsome, and thank you for the raid again, Zeddy. Guys, if you're not checking out Looking Cool Zedman, you're not doing left right, please go follow Zedman. Absolute gold mine of a friendo. We love you, Zeddy. Sleep well, Zeddy. Sleep well. So after the female eats the male's head, after you know what, he's still alive. Yep. With fruit flies, if you chop off their head, they're alive for 24 hours. Hi, Freshie! I'm glad you're doing all right, Freshie. We'll give you your fancy pants shout out in just a moment as well, Freshie. Zedman, sleep well. Thank you for the raid, and know you're breathtaking. I'll talk to you tomorrow morning, as we always do. So much science sounds the same, but really isn't. Ganglion exists, and it's, yep. It's all over, Clement. It is all over of connections of it. And yeah, Dame Karen, nope. We're going to nerf that comment, Freshie. Don't you dare, Freshie. Don't you dare, Freshie. Yeah, Dame Karen, with the fruit flies, if you chop off their heads, the males will mate with a headless female. And the headless female will lay eggs up to 19 hours after being decapitated. It's quite wild, actually.
Remembers locations of food sources and hives, regulates communication, and even coordinates navigation over huge distances. And this vast diversity of behaviors is controlled by an organ the size of the head of a pin, with less than 1 million neurons compared to our 86 billion.
Yep, I mean, Dame Karen, you don't need a head to mate for fruit flies. Actually, there is a mating dance, Dame Karen, that flies do. And there is a visual component to it. But the visual component tends to be on the part of the female deciding. So the female, when she has eyes, she can choose whether or not to mate with a male. And so the male actually will shake his wings towards the female, and you can see different color patterns on the wings, and the females can see like this unique component. And the females can then choose if they want to mate or not. If they are headless, the males still do the dance, but that's about it. Like they don't. There's no actual mate rejection because the females, the mating is olfactory, visual, and auditory. They can still hear, but no, it's not the same. So then the males just mate, and it's quite fascinating that the males are so discerning that they don't even care if you're headless or not. Guys, go check out Freshie, the game dev extraordinaire. We love you, Freshie. We love you, Freshie. No, Dame Karen. The males don't decapitate her. This is a scientist doing it. So you take micro scissors and you cut off a head. This is naturally in the wild. It does not happen. This only happens in an experimental setup. And we ask that. No, I mean, John Cheese, I'm not gendering what the scientist is. Absolutely not. Absolutely not. It will not chop off the head. So female, like fruit flies aren't strong enough to rip off each other's heads. So that's just that's a human intervention there.
But even though the insect brain is organized very differently from ours, there are some striking similarities. For example, most insects have smell detectors on their antennae. similar to those found in human noses. And our primary olfactory brain regions look and function rather similarly, with clusters of neurons activated and deactivated in precise timing to code for specific scents. Scientists have been astonished by these similarities because insects and humans are not very closely related. In fact, our last common ancestor was a simple worm-like creature that lived more than 500 million years ago. So how did we end up with such similar brain structures when our evolution took almost entirely different paths? Scientists call this phenomenon convergent evolution. It's the same principle behind birds, bats, and bees separately evolving wings.
So Cobra, it might be, but the female's going to die very rapidly after, so she'll live 24 hours. But Cobra, when a fly mates off one mating, she can lay eggs for up to a week. So it's in the male's interest that the female stays alive for longer because his genetic information will be passed on more. Is there an insect that can survive without a head for a really long time? Sooner or later, they have to starve to death. Yep. Actually, Clement, fascinatingly enough, there are parasitic wasps that are hatched without a mouth. They don't eat as an adult. And they live for about a week. They mate, they lay eggs, and they die. And they survive off the fat from, like, as the larval stage they have when they undergo metamorphosis. Yep, same Karen. The female fruit fly can live about 24 hours, lay eggs, and mate without a head because it's a decentralized nervous system. But again, for the male, like, the male wants her to stay alive longer because then she'll lay more eggs that were fertilized by his sperm, which means that it's a better, I guess, evolutionary success for that male, if that makes sense. And yeah, Clement, I'd say about a week, because if it's not headless, but they don't have a mouth, so sort of thought everything had to have some sort of eating system. Well, so Clement, but insects are different, right? Because they have an egg, larva, pupa, and adult stage. And imagine like in your larval, if you're an insect, if you eat enough in your larval stage and you have enough fat reserve that when you undergo metamorphosis and you become an adult, then you still live. You still live with no issue. That's pretty cool. So I think it's different than our views on like mammals, for example, right?
Similar selective pressures can cause natural selection to favor the same evolutionary strategy in species with vastly different evolutionary pasts. By studying the comparison between insect and human brains, scientists can thus understand which of our brain functions are unique and which are general solutions to evolutionary problems.
It is crazy as
But this
ferret.
is not the only reason scientists are fascinated by insect brains. Their small size and simplicity makes it easier to understand exactly how neurons work together in the brain. This is also valuable for engineers, who study the insect brain to help design control systems for everything from self-flying airplanes to tiny search and rescue roach bots. So, size and complexity are not always the most impressive things. The next time you try to swat a fly, take a moment to marvel at the efficiency of its tiny nervous system as it outsmarts your fancy brain.
So Dame Karen, I think that's a much warmer and fun place to end, right? Like, it's not as dark as a moment ago when it was like, all right, we as humans are totally nerfed because of everything that wrong can happen with us and our brains are deteriorating. This is a little bit more hopeful. This is saying, insects are very cool. They're very smart. There's a lot of different things that they can teach us. And by the way, we might be able to utilize them to probe their genetics and their neurobiology and learn more about how things work, which is pretty cool. Is this in a lesser state of how? Not quite. So Twitch Hag, chickens have a centralized nervous system. The running around without a head is a reactive response. It is just like final neurons just signaling. and like overfiring, and then it's just you're gone. Versus the decentralized nervous system, there's no spinal cord. There's information coming from other neurons elsewhere in the body, so you don't have to have continuous signaling from the brain to do your standard behavior. Versus like for us and chickens too, like it's a quick running reaction, but then you're gone. So it's not a sustainability question anymore. How long? I am not sure, Dane Karen. I'm not going to Google it when we're live either, because I figure that will be a horrifying rabbit hole that we might plug up that rabbit hole and not go down that one. Happens to humans as well. I would imagine so, Clement, depending on certain situations. But it's, again, a decentralized nervous system versus centralized is the differences there. All right, chat. My mods are reminding me that, Balint, tomorrow you have to get up really early because you have a child. You have a baby.